Hello everyone, I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Achieving Cell-Free Gene Expression with Acoustic Liquid Handler, and our sponsor is Beckman Coulter Life Sciences. Our panelists for today are Dr. Vincent Noireaux, a professor at the University of Minnesota, and Aset Khakimzhan, a PhD student at the University of Minnesota. You can type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Noireaux. Please go ahead.
Thank you, Christy. My name is Vincent Noireaux. I am a professor at the University of Minnesota, and I will present this webinar with Aset Khakimzhan, a PhD student in my laboratory. In this webinar, we will cover the four following bullet points. First, I will introduce cell-free gene expression, a rather new technology to do some synthetic biology and bioengineering. I will then introduce the Echo liquid handler from Beckman Coulter Life Sciences. Aset will cover the third bullet point on prototyping with TXTL, and we will conclude this webinar with some suggested literature. Let me start with some terminology. There are many different ways to define the in vitro synthesis of proteins from DNA. CFE for cell-free gene expression, TXTL for cell-free transcription translation, IVTT for in vitro transcription translation, and CFES for cell-free protein synthesis. As described in the paragraph below, cell-free transcription translation is becoming a popular laboratory research tool in biology, bioengineering, synthetic biology, and biological physics. Major applications of TXTL include biomanufacturing proteins and prototyping DNAs, from regulatory elements to gene circuits. Combined with automated liquid dispensing, TXTL can dramatically accelerate synthetic biology applications. Beckman, the sponsor of this webinar, provides a unique automated acoustic liquid handler ideal for TXTL work. In this webinar, we will use CFE and TXTL to define the in vitro synthesis of proteins from DNA. In this first part, I will introduce cell-free gene expression, what it does, how it is prepared, and what scope of application it covers. Synthetic biology environments, there are actually three of them, in vivo, in silico, and in vitro. Synthetic biology, it's programming with DNA to direct and control biological systems, to understand gene regulation and make simple computations, to use them either as molecular scale factories, and to create new materials. In this webinar, we will focus on synthetic biology application in vitro, using cell-free gene expression as an experimental platform. Cell-free gene expression systems are prepared from living cells. Typically, the first step consists of choosing a type of cells. In many cases, it's Escherichia coli. The cells are grown in a culture flask, and a lysate is prepared by breaking the cells and isolating the liquid part of the cell or the cytoplasm. This lysate contains the molecular machineries to perform transcription and translation in vitro. The cell-free gene expression system is also composed of other solutions, an energy mix containing the ribonucleosides, such as ATP and GTP for the energy, and an amino acid mixture that contains the 20 canonical amino acids, and other cofactors, such as molecular coders, like PEG or FICOL, and of course, your DNA prepared using standard procedures. Cell-free gene expression reactions are performed in different types of volumes. It can be performed in flask from the milliliter to the liter scale. It can be performed on well plates or test tubes and using a liquid dispensing machine like you are going to see in this webinar. In that case, the cell-free reactions have a volume ranging from the microliter to a few tens of microliters. And it can be also done on microfluidic chips and synthetic cells. In that case, the cell-free reactions have a volume ranging from the femtoliter to the microliter scale. What are the advantages of cell-free synthetic biology using TXTL systems? The first one is the speed. Cell-free gene expression is a very fast experimental environment. The setup of the reaction is fast. It's compatible with high throughput equipment, as you are going to see in this webinar. And it's very useful to prototype very rapidly DNA parts and DNA circuits. Parts, it takes just a few hours, and gene circuits can be prototyped, or if you like, characterized in a couple of days, compared to weeks using a living chassis. The second advantage of cell-free synthetic biology, it is its safety. A cell-free expression system does not contain any living cells, it does not reproduce, it does not expand. The typical cell-free gene expression systems are based on standard E. coli strains, and cell-free expression systems have become, in the last 10 years, easy to use. Finally, the third main advantage of cell-free gene expression system for synthetic biology is its quantitative aspect. You can calibrate the fluorescence of your devices, you can control the DNA stoichiometry, you can develop models, and you will see that there is a good agreement between the results obtained in cell-free gene expression reactions with the results obtained in vivo chassis like E. coli. Here is an overview of the TXTL applications. You see on top and on the left, you choose your vessels. It can be test tubes or well plates. You prepare your DNA, you clone your DNA and prepare it, and you use your TXTL mix, and you incubate. Depending on the kit that you use, it can be just a few hours, up to 20 hours, at temperature typically between 29 Celsius to 37 Celsius. And after this incubation time, you get your product or your application. What is the scope of application of cell-free gene expression? The first one is prototyping biomanufacturing. You can prototype and characterize DNAs very quickly in a TXTL system from parts to circuits. You can prototype enzymatic pathways. You can also biomanufacture. You can produce peptides or enzymes very quickly, like antibodies, pharmaceuticals, and you can also perform unnatural chemistries, like, for example, using a non-natural amino acid that you are going to insert into your product, a peptide or a protein. The second application of TXTL is self-assembly nanotechnology. You can produce, for example, virus-like particles. You can produce phage parts. You can produce all phages in test tubes. You can do some directed self-assembly and some new self-assembly. As you can see, just below this paragraph, it is an electron microscopy image of the phage T7 entirely expressed and synthesized in a cell-free gene expression system performed in a test tube. And finally, the third main application of TXTL is synthetic cells and material sciences. TXTL is very well suited to build synthetic cell systems, like you can see in the fluorescent image at the bottom here, at the bottom right corner. You see two liposomes loaded with a cell-free gene expression reaction to express a cytoskeletal protein, the cytoskeletal protein MREB, that polymerizes at the inner membrane of liposomes. With this system and TXTL, you can study cell mechanics, you can develop diagnostics, you can develop microchips, you can build artificial cells by incorporating non-natural molecules, and you can develop biotic-abiotic interfaces. A liquid handling machine like the Echo from Beckman Coulter Life Sciences is particularly well adapted for two of these applications, and especially prototyping biomanufacturing, as you are going to see in a few minutes. It is also useful for self-assembly nanotechnology. In this webinar, we will present some results obtained using the kit MyTXTL sold by Arbor Biosciences. MyTXTL was specifically developed for synthetic biology application. It has a number of characteristics. It is very simple to handle. It has a very high performance at the level of 2 to 3 milligrams per ml of protein produced. It has some very particular capabilities. It can produce proteins from 10 kilodalton to 200 kilodalton. It has a great versatility. It's compatible with a lot of different promoters. It has a very fast turnaround and an all-time flexibility. In the second part of the webinar, I will introduce and present the Echo liquid handlers from Beckman Coulter Life Sciences. There are two Echo liquid handlers from Beckman Coulter Life Sciences, the Echo 525 and the Echo 650. The main difference between the two liquid handlers is the volume of the droplet. It is 25 nanoliters in the case of the Echo 525 and 2.5 nanoliters in the case of the Echo 650. The speed of dispensing for the Echo 525 is up to 6 microliters per second, while it is up to 1 microliter per second for the Echo 650. The Echo 525 is ideal for reaction of around 10 microliters, below, a little bit below and above, and the Echo 650 is ideal for reaction of the microliter scale. It can be sub-microliter up to a few microliters. The transfer in the case of the Echo 525 is done from a 384 well plate, a source plate, and the destination plate can be of any kind in terms of number of wells, from 6 wells up to 384 wells. In the case of the Echo 650, the transfer, the source plate can be either a 384 or a 1536 well plate, and it can dispense in well plates from 6 well up to plates that have 1536 wells. In this slide, you see the technology of the Echo sold by Beckman Coulter Life Sciences. On the top left corner, you see a source plate loaded with 2 different solutions. One is purple and one is yellow. And below the purple solution, you see the acoustic system that is going to produce small droplets that are shot above into the destination plate that is upside down just above the source plate, as you can see in the picture on the right here. You see the source plate with all of the solution of different colors, purple and yellow, and you see the destination plate white just above upside down. In the time-lapse sequence that you see at the bottom of the slide, you see the formation of the small droplets that are shot in the destination plate during the dispensing. How the Echo liquid handling works. It works based on acoustic energy, as you have seen just before, with droplet of 2.5 nanoliters for the Echo 650 or 25 nanoliters for the Echo 525. It has a high accuracy and precision, a very low energy transfer, and it preserves the sample integrity. It is rapid, up to 6 microliters per second for the Echo 525 and up to 1 microliter per second for the Echo 650. And it can do any well to any well. You can see this on the right here. You see at the bottom the source plate with 4 different solutions labeled as A, B, C, D. And just above, upside down, is the destination plate where you dispense. Any combination of the solutions A, B, C, D can be dispensed in any well of the destination plate. As you can see here in the destination plate, we have 4 different wells with 4 different volumes of the 4 different solutions, but keep in mind that you can absolutely mix and at whatever proportion the different type of fluids that you put on the source plate. The dynamic of fluid analysis, you can adjust the transfer parameter in real time for a variety of fluids. It is, for example, absolutely compatible with biological fluids like a cell-free gene expression system. You can add various samples and reagents at different concentrations without any user calibration. And it's highly compatible with DNA solution, RNA solution, enzymes, master mixes in glycerol storage solution, for example. This, it concludes the second part of the webinar, and I will give it to Aset to introduce and discuss some application with cell-free gene expression and the Echo liquid handler.
All right. Thank you, Vincent. So I'm going to talk how we use an Echo liquid handler from Beckman Coulter Life Sciences to prototype TXTL circuits and genes in TXTL. So whenever we prototype something in TXTL, we have to first consider for, let's say, for example, for a single gene, what is the promoter of a gene? What is the untranslated region for the whole plasmid or the linear DNA? What is the specifications of a ribosomal binding site? And what is a terminator for the gene? And then when we look at higher scales, let's say building genetic circuits, when we have 5 to 10 genes in TXTL, we would be interested in prototyping the regulatory components of the circuit. So right now we have riboregulators, RNA aptamers. Recently we've been using a lot of CRISPR to regulate circuits. And then, of course, the transcriptional factors of, that we've been using for a while. And whenever we express genes in TXTL, we would use either linear DNA, which we can order as G-blocks, or we can PCR amplify. We would use the traditional plasmids that are used always in synthetic biology. If we're reading out a genetic circuit, then we would always have some kind of gene that would report the fluorescence. And depending on the fluorescence of the reaction, we can tell if the circuit is working or not. Or if we're interested in a metabolic pathway, we can measure the luminescence of a reaction. So the advantages is that cell-free has a great agreement between itself and in vivo results. And in addition to all of this, this is very fast. This happens quickly in TXTL. We can measure and acquire results within days for single circuits, and within a day for a single gene, and in a couple of days for circuits. Traditionally, or I guess before we had an Echo liquid handler, we would do reactions by hand. So reactions by hand would always be 10 to 12 microliters. And we would have to mix the lysate, so it's the TXTL, the main components, the nutrients that would be used to produce the proteins or the RNA. We would introduce the DNA, which would be the instruction, what genes we want to, what proteins we want to make, so we would put the DNA with the genes. And then to balance everything out, we would put in some water. And so here in the picture, you can see there's a tube with about 12 microliters of TXTL. And here we are producing EGFB, and so we input the gene P70 EGFB, and as you can see, the reaction is green. However, when we do reactions by hand, we would have to do everything with a pipette. Unfortunately, pipettes have larger errors, especially when we work with smaller volumes. It's you only can have one pipette in your hands, so it's unlikely to do high free output reactions. again, you do it with your hands. It's difficult. If you're doing it for a while, it can get tiring, and there's definitely is a human physical limit. It takes a lot of time. So to set up a reaction, to think it through, to pipette everything, it's a lot of work. And at the end of the day, for each reaction, we have to use 10 to 12 microliters of a TXTL to acquire just one data point. Now, once we use Echo 550 or 650 by Beckman Coulter Life Sciences, everything becomes much easier. So we have the lysate, we have the nutrients, we have the DNA, we have the water, all the same components that we had by hand. But instead, we don't have to do it with a pipette. The Echo would dispense everything for us. So since the dispensing is automatic and it's a robot, the errors are small. And again, it's a robot. Everything happens the same way. If you program it, it's going to work the same way. So it's highly reproducible. It's designed to be high free output. You don't have to do anything with your hands other than load the source plate. And so there really is no physical limit. It's 5 to 10 times faster because of that compared to doing everything by hand. And now we can do reactions that are much smaller than the 10 to 12 microliters of the by hand reactions. We can go down to 0.5 microliters. And in addition to all of that, the liquid handler accepts CSV files as their instruction. And so if you want to semi-automate the design of your experiments, that can easily be done. We use Python to make the CSV files, and then we put them onto the Echo software, and it performs the reactions. But it can be done with any language that can output CSV files. So basically, any language can be used to design your reactions and make it faster. And here is a typical output of a 96 plate well plate. And so there's 8 rows, 12 columns, and you can see for each of the cells, each of these squares, there's a different curve. So the x-axis for all these curves is time, the y-axis is the intensity of the fluorescence, and you can see there's different reactions, and all of this can be done within 15 minutes with an Echo. Doing reactions by hand or doing reactions with an Echo, you can see there's a huge difference. So if you're doing the reaction for the first time, that means you haven't been doing this reaction before, and it's the first experiment, then the Echo is about 5 times faster than doing the experiment by hand. So with the Echo, you have to program the Echo. You would have to put the liquids into the source plate, and then put it into the machine, and from there the Echo would dispense it into the destination plate. And that is very fast, and by hand it's just pipetting, and it takes a while. If, however, we're repeating the experiment, so pretty much the same experiment, that means we wouldn't have to program it again on the Echo because it's already programmed. So all we have to do is load the source plate and just put it into the Echo, and it goes. But by hand, it barely gets faster if you're repeating the experiment because, again, you still have to pipette the whole time. And so repeated experiments 10 times faster with an Echo, and if it's your first experiment, then 5 times faster with an Echo. So that's a huge benefit. So I just wanted to present a couple of examples of how an Echo can be used in a cell-free synthetic biology lab. So here's this Echo 525 by Beckman Coulter Life Sciences. So that's the one that has a resolution of 25 microliters. Each droplet is 25 microliters. And a colleague lab of ours, Paul Freeman's lab, they were interested in designing new cell-free systems. So can you make a cell-free system from a non-model organism? Because right now, cell-free systems, the most popular one that we use is based on E. coli, but there's also Pure, which is completely synthetic. But what about using other organisms? And here they have this paper published by, it's written by Moore et al. In the, again, Paul Freeman lab. And one of the parts of when you're making a new self-read system is having to check your regulatory elements. So in here, the way they use the Echo is they designed a lot of different ribosomal binding sites, and they all had different spacer lengths, so AT spacer lengths of the ribosomal binding site. And when you're designing a new system, you want to find out which ribosomal binding site should you use and how, what kind of ribosomal binding sites should they put on their regulatory, on their genes. And so here you can see they have their Echo, they upload, they make a plate, put it into a fluorescence reading machine, and they get the output. And based on this output, they can see which of the ribosomal binding sites work well. And there's the x-axis where it's the length of the spacer, and the y-axis is the GFP fluorescence. And based on this, you can decide which ribosomal binding sites you should use. And so the Echo really helped make the whole process faster. So this is how the Echo 525 can be used. Just one example. And here's an example of how we use Echo 650. 650 is the one that has 2.5 nanoliter droplets. Again, we have our lysate, we have our DNA, we have our resources, we have our water. We put it all into the source plate, and the Echo dispenses that. It takes 15 to half an hour, 15 minutes to half an hour. It can be less if we're not doing a whole plate, which happens every now and then. But it would never exceed 30 minutes for us in our lab. Then we put it into the fluorescence reader to get the fluorescence for each reaction, and that takes 3 to 12 hours. So in this case, I wanted to look at a negative feedback loop. So here you can see there's 4 genes. So if you consider having 4 genes in a circuit, that means that you have to make sure that the concentrations of these 4 genes is correct for the circuit to work. And so 4 genes, that means for each of a gene, you have to change the concentration and see how it would work with the other ones. So that's a lot of complexity. And then if we study even bigger circuits that have more genes, the complexity increases even more, and you have to scan the whole parameter space. And imagine doing it by hand, or imagine doing it with an Echo. So Echo, again, 5 to 10 times faster than by hand. So scanning the whole parameter space would be 5 to 10 times faster with an Echo than by hand. So that's a huge benefit. In addition to that, you can see that on the bottom right corner of the slide, we have a comparison of hand and Echo. This is the absolute expression of a reaction. And you can see that by hand and by Echo, the qualitative side of a plot is the same. So hand low, Echo high, hand low, Echo low, hand high, Echo high. And so it's pretty much the same thing, but for Echo, we get consistently higher expression, which is really great because it helps us read out more. And if we're working with a circuit that doesn't have too much expression, having just slightly more expression helps. And these are the examples for using Echoes for TXTL. And with that, I'll finish it with the suggested literature. And yeah.
Thank you, Aset, for presenting a few examples of how to use the Echo Liquid Handler from Beckman Coulter Life Sciences with cell-free gene expression systems. In this slide, we propose some literature about the E. coli cell-free expression system that was presented in this webinar, some literature about cell-free gene expression in general, 2 review articles if you want to know more about cell-free gene expression, and some articles using both cell-free gene expression and the Echo Liquid Handler from Beckman Coulter Life Sciences. So the work done and the results that you have seen in this work were obtained by graduate students and postdocs in the laboratory of Vincent at the University of Minnesota. You have on this slide some contact information if you want to have more information. And the results presented in this webinar were sponsored by the following agencies.
Thank you, Vincent and Aset. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. And joining us for the Q&A today is Sohaila Beck, Commercial Product Manager for Genomics at Beckman Coulter Life Sciences. Okay, we'll now start the Q&A. Forgive me, folks, I was on mute. Let's go with our first question for both Vincent and Aset. Vincent, maybe you want to take this one first. How does the liquid not fall down while it's inverted in the destination stage?
Yes, so it's a very important question. So of course, we dispense, when we use a cell-free gene expression system, many different types of solutions into either a 96 well plate or a 384 well plate. And it turns out that capillarity is just enough to keep the liquid not falling from the plate. For a typical cell-free gene expression reaction that we do in our laboratory, the reaction volume is on the order of 2 to 3 microliters. And at that scale, capillarity in the wells is absolutely enough to keep the liquid in the well without any problem. And for any type of solutions, they are all aqueous solutions, water-based solutions, either for the extract containing a lot of proteins, or the energy mix containing a lot of small molecules, or the amino acid mix containing the 20 amino acids, or molecular crowder, for example, the PEG solution, relatively viscous. All of those solutions are behaving very well, and there is no problem of liquid falling after they are dispensed in the plate, the destination plate that is upside down. That's it. Maybe Aset, you want to add something about this?
Yeah, we experienced no problems with the droplets falling, and yeah, we can go up to volumes of 5 microliters, and yeah, I never worry about that.
Great. And does the viscosity of the liquid affect the delivery by Echo?
Well, that's a very good question. The viscosity of the solution that you dispense does not really affect the delivery by the Echo, but the Echo is already incorporating this aspect into the dispensing. It's just, I think, a question of choosing the type of liquid that you dispense, and especially at the level of the viscosity. And then the Echo is going to adjust the dispensing. So there is no problem at that level. For example, using the Echo 650, the PEG solution that emulates molecular crowding to enhance cell-free gene expression is relatively viscous, and it's not a problem at all. Maybe Aset, you want to add something, and maybe Sohaila also, you may have a word about it.
Yeah, sometimes I guess if something's more viscous, the regular mode that we use for 650, which is GP, doesn't always dispense, but then we can switch to other modes that the Echo has. So depending on what liquid you use, you can switch to the different modes of shooting the liquid, and it always works.
Yeah, I agree with that. This is Sohaila. So we have different calibrations. We can dispense up to 50% glycerol with the GP calibration, and there is another very universal calibration called CP, which transfers slower, but it can transfer a very wide range of liquid. Thank you.
And Sohaila, I believe the next question is for you. Does Echo have any temperature controls when samples are being deposited on the plate? And are TXTL components stable at room temperature? And if so, for how long?
I'm going to take the first one and pass the second one in terms of the TXTL reagents being stable or not to Aset and Vincent. Echo currently works at room temperature. There are a lot of genomics reactions, NGS library preps, and I am not as diverse knowledgeable on the TXTL part and reagents if they are needing to be at different temperature, but the reactions are very stable and successful. Reagents can be used over and over. So we haven't had a wide need to change the temperature, but Echo works at room temperature currently. Aset and Vincent, may I pass the second part to you?
Yes, thank you, Sohaila, and I will have Aset also say a word on this. It's a very important question, very good question and important about the solutions, the TXTL solutions. So typically you have it depends on the TXTL system that you use, but typically you have a mix, which is the extract containing the ribosomes and all the proteins necessary for transcription and translation. You have another mix, which is the energy mix for ATP regeneration, another mix with the amino acids, and so on. So those solutions are biological solutions. And it turns out that when we use the Echo to dispense our reactions, it takes for us about 10-15 minutes to cover a 96 well plate. With reactions, the final volume of the cell-free reactions are on the order of 2 to 3 microliters. And in 15 minutes, at room temperature during dispensing, there is no problem at all. All of those solutions are perfectly stable. We even sometimes and often recover those solutions after dispensing because we can use them again. So no problem at all at that level. But maybe Aset can be more detailed about this.
Yeah, I agree. So for one plate, there's definitely no problem. And sometimes if we want to do multiple plates, that takes more time. And if I'm trying to use the same stock of the extract lysate, then I will put it on ice. But that's more of a safety precaution. I don't think it helps. It's just sort of lowers my anxiety regarding the possibility of the extract deteriorating over time. But yeah, I would say the extract is probably the biggest, the first thing to go. Everything else is pretty stable.
Great. So Aset, could you tell us what the typical volumes are that are transferred and then dispensed in the destination plates?
Usually we will do 2 microliters. That is our standard. That's like 95% of experiments are even more. We do 2 microliters. Sometimes we go down to 0.5 at first, perfectly well. We still get very good expression with our benchmark circuits. And then we go up to 5 microliters if we want more volume. And that also works very well. We just have to adjust for the oxygen intake of the reaction. But so we have to play around with the parameters of the reaction. But that's a very small thing to do. And so it always works, yeah. So from 0.5 to 5 is easy, and I'm sure we can go down to even less or more. It's just for less, the resolution becomes an issue sometimes. So I guess we could do maybe 25 microliters, and I've heard that some experiments go even down to very small volumes. I've seen some work on that. So yeah.
And Vincent, a question for you. Could you tell us what you think is the future of TXTL or CFE in applied science or biopharma? Do you see this technology being used in biological drug discovery?
Absolutely. So it's a very broad question. I will try to be brief. So TXTL, or if you like, cell-free gene expression right now, is attracting a lot of interest. there are more and more laboratories using cell-free gene expression, and for a number of applications that is also increasing every year. If you look at some certain reviews published recently, they report the number of articles based on cell-free gene expression, and it's literally going up every year. So the future of TXTL seems to be very bright. And there are, once again, new applications every year. For example, in the past 4 or 5 years, TXTL has been shown to be ideal for many diagnostics, biomedical diagnostics. It's very compatible with this research area. Do you see this technology being used in biological drug discovery? Absolutely, and there are already some companies using cell-free gene expression to prototype certain biologics like antibodies, for example. Cell-free gene expression system can be used to synthesize antibodies, and some companies are already using this and producing antibodies for personalized medicine. So to summarize, cell-free gene expression is an incredibly versatile technology with already currently an impressive number of applications, and new applications are demonstrated every year. So cell-free gene expression is definitely very trendy, and I have a little bit of experience with this. I think that's going to continue for many years because there are a lot of discoveries, a lot of areas or fields that have not been explored with TXTL, and the potential is huge. That's it.
And what do you think are the next important improvements that need to be made to expand the current cell-free expression capabilities?
So cell-free gene expression systems are already capable of a lot at the level of the yield. The yield has become interesting. Cell-free gene expression system now produce protein at relevant biological yields for biomanufacturing. I would say potential improvement is at the level of what type of protein you can produce. Cell-free gene expression system can produce a lot of proteins, but not all the proteins that are produced in living cells. There are still some progress to make at the level of, for example, proteins containing post-translation modification, like disulfide bonds or glycosylations. Although in the past year or two, very important progress has been made at that level, but still there are still some room for improvement for what we call complex proteins containing PTMs, post-translation modification. Disulfide bonds, although we start doing this, the community specialized in cell-free gene expression system knows how to do this now. Post-translation modification like glycosylation, there are still some clear improvement to make, but the community is working with that on this and developing new cell-free gene expression system to be able to produce complex proteins at least 1 milligram per ml. We are converging towards this, and I'm sure that in 1 or 2 years, such biomanufacturing of complex proteins will be possible. A lot of work also is done in extending the properties, expanding the properties of cell-free gene expression systems so that you can do non-natural biochemistries. And one example is the introduction of non-natural amino acids into proteins. And here also in the past couple of years, some major improvement and progress have been made. It's possible now to synthesize proteins in a cell-free gene expression system and include several different non-natural amino acids that are used after for specific chemistries or to literally enhance the properties of a protein. So once again here, the possible improvements are many, I would say. And the community right now is working on this, and you will see in the next few years, I'm sure, some very important improvement, not only at the level of the yield, but at the level of what type of proteins you can synthesize.
Great, thank you very much. I'm showing no other questions in the queue right now, so I'm going to wrap it up there. We'd like to thank Vincent Noireaux, Aset Khakimzhan, Sohaila Beck, and our sponsor, Beckman Coulter Life Sciences. Please be sure to join us on November 4th for part 2 in this series, where we'll cover the characterization of CRISPR technologies using TXTL and the Echo liquid handler. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. And if you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this genome webinar.